Yes, I mean, it's been a very busy year for us, and many of you will have seen quite a change in the organization, not just myself, but throughout the ranks. But I wanted to take the opportunity to quickly introduce our new Director of Business Engagement and Operations, Liz Fletcher. Liz joined us in March, and so she'll be working alongside Ian Archer, our Technical Director, and myself, and the rest of the team in helping to shape the centre's strategy and deliver the main objectives that we have for Scotland and for the economy and for all of you to support your innovation journeys and accelerate commercialization. Liz, do you want to just come on to camera and say hello?
Yep, say hello everybody. It's a shame we're not there in person, but perhaps one day soon. I'm really looking forward to getting to know those of you I don't know already. But if you would like to have a chat, please do reach out to me. I'll put my email in the chat. So look forward to getting to know you better. Thanks, Mark.
Great, so we're delighted Liz has joined us. So, you know, this is our member event. So I ideally would have liked to have this, you know, as our face-to-face and rub shoulders with all of you and have a good chat over the evening, but sadly, of course, we're unable to do that. So hopefully this suffices at the moment, and we'll hopefully look forward to seeing you all in person. Just in terms of the membership, over the last year, we've welcomed an additional 20 new members into the organisation, which is brilliant, and we've met lots of different people from different sectors and different backgrounds, and it's been really fascinating for all of the team. I guess in particular, I would just flag a welcome to Victrex and Beckman Coulter, who are our two most recent joiners, and I look forward to getting to speak to you in the near future. The other thing that you will have picked up on, or if you haven't, it will drop in reasonably soon as renewals start to kick in, is we have had a good look at the membership model and taken on board a lot of the comments that many of you who interact strongly with us made. And so we have now generated a more simplified model, and it will come into effect on the 1st of August for the existing members. And also, if you have any queries about that, then obviously speak to Caroline Cuny. That would be your main point of contact on that. Critically as well, we've decided to go for a single renewal point as well. So that'll be the 1st of August, just to simplify everything rather than rolling quarterly renewals. I mean, one of the main reasons that you interact with us, of course, is on funding and the network and helping innovation to grow in Scotland in this space. But don't forget that there are usually funding calls running, and our innovation funding can fund up to £100,000. And actually the deadline for it's the 1st of July. So for those of you not yet working on it, you might want to engage with the team on that. And then we also have the Scale Up Accelerator project call for SMEs, which is an open call with no closing date. So as and when you feel ready for that, please come and interact with us. Positively as well, we're launching 2 funds today. We were launching a feasibility fund, which is up to 20k for proof of concept projects between companies and academia, and also a brand new spin-out fund, which is up to 20k towards university costs for spin-out projects. And our team will be very happy to talk to you about any of those, of course. As well as our innovation support, clearly we do a lot around skills and workforce development. And I wanted to say a thank you to all of you that were able to take a placement of our master's students this year. We very much appreciate it because of the uncertainty. I mean, the Green Skills agenda is clearly on the page, not just within Scotland, but globally, and is seen as incredibly important in the post-pandemic recovery. Also, we announced recently the last cohort of PhD studentships who've been allocated to businesses as well. And there are still some opportunities available through the IBioIC CTP website, which you'll find at IBioICCTP.com. So lots going on, both in skills and innovation. But thinking about where we're going at the minute and things that have happened that you may not be aware of, you know, in lockdown, we did pivot activities, as did many organisations during the challenges of the pandemic. and we pivoted, you know, IB core functions and skills to support, you know, medicines, supply chains. We're more interested now as well in looking at that further. We're supporting the growth of manufacturing in Scotland. You might have picked up actually, you know, work that we've been doing on field studies for a reintroduction of sugar beet into Scotland as an energy crop and as a sugar supply for potential expansion for obviously fermentation-based industries in Scotland. And interestingly, it was picked up by the Times on Monday as one of their featured articles, which was really good to see that at a national level across the UK, you know, we're being seen as leading in some of this space. COP26, we've decided we will go ahead to host our own event within the programme of the fortnight, and so we'll be hosting an event which you will all receive invitations for, your organisations will receive invitations for, and it'll be held at the TIC in Glasgow, providing we can at that point, you know, be able to have some more face-to-face time. We're also working with colleagues in Scottish Government around COP26 and also Scottish Enterprise and Scottish Funding Council around what we might do to join them to show what we're doing in Scotland. So really positive, great opportunity, and we're delighted to be working with partners on that. The other thing which we've just kicked off is an update on the National Plan for Industrial Biotechnology for Scotland, which we will aim to release the update in 2022 at the IBioIC conference. So this is really the document that we are measured against in terms of growth for Scotland, economic, our company turnover figures, and also the numbers of businesses using industrial biotechnology. So I look forward to interacting with some of you on that as well, and showing what ambition we've really got for Scotland to grow its bioeconomy supply chains. So on to the main part of today. I hope you felt that the agenda was suitably different than the last time, and this gives you an opportunity to hear a little bit about what Scottish Government are doing, and also, you know, some great showcases that you'll get lots of information in a very short space of time, and give you an opportunity to think it through and reach out to some of those people, hopefully for networking. So firstly, I'm delighted to welcome our first two speakers for today, Heather Perman, who's Policy Advisor, and Ali Morish, who's Policy Officer. Both work in the UK Industrial Strategy and Technologies team for Scottish Government. And they're going to talk a little bit about their team and what the main priorities are in relation to industrial biotechnology. So I'm delighted to have been working closely with Heather and Ali over the last 12 to 15 months. And so I'm delighted that they're able to join us. So welcome, Heather and Ali.
Hi, thank you so much. Hi, everybody. I'm delighted to be here on what looks like to be an interesting and engaging afternoon. Well, firstly, thank you to Mark and Debbie for inviting us both along to talk today. We've been having lots of discussions with them about how to reach out to the membership, and this seems like a perfect opportunity to say hello and explain who we are. The theme for our session is working in economic development. So we're going to cover a bit about our team and our priorities and tell you a little bit about our wider division too. So we work in the Directorate of Economic Development within Scottish Government, working in Manufacturing and Industries Division in a team called UK Industrial Strategy and Technologies. And the technology that we look after, of course, is industrial biotechnology, which has led us to lots of conversations with IBioIC and, of course, many of you on here today. So we've both arrived at this role in slightly different routes, and we thought we'd explain where we both come from. So I'm Heather Perman. I joined Scottish Government in April last year during the first lockdown, so never actually having made it into the office, unfortunately. So I joined from the private sector, having worked in the banking industry for 14 years, specialising in change management. So I decided to change my career, and that led me to the civil service and to industrial biotechnology, which I'll be honest, I had to Google in preparation for the interview, so no background in it whatsoever.
Hi, I'm Ali Morish. I joined the team a few months before Heather did, and I was also new to Scottish Government and to industrial biotechnology. I was fairly quickly redeployed onto our COVID response before returning to IB in June last year. Before the Scottish Government, I worked as an electrical engineer in the automotive industry and at a printing services startup. And before that, I was a sailing coach for some of our national squads. So this is quite a change of environment. So we have both had to learn about the sector while being entirely home-based during what has obviously been a challenging year for us all. But what has really struck us about working in this policy area is how passionate and excited everybody is about the ideas, innovation, businesses, and the opportunities and potential for Scotland. And it's really infectious. The guys at IBioIC have made it easy for us to learn all about IB and have put up with lots and lots of questions from us, and it's really made our jobs much easier.
So what we thought might be useful is to cover what our role actually entails, a bit about what does actually working a policy mean and how do we spend our days. A big priority for us within our role is on the National Performance Framework and National Outcomes, which describes the Scotland we want to create, a more successful country which gives opportunities to all people living in Scotland with enhanced well-being, reduced inequalities and a sustainable and inclusive economy. A key part to our role is all about fact-finding and essentially storytelling. What's going on in Scotland? Who are the key stakeholders? What are the challenges and potential opportunities? So we do that through desktop research, speaking to stakeholders like yourselves, individual companies, IBioIC, the enterprise agency, Zero Waste Scotland, keeping watch for news, articles, funding calls, announcements. And it's all about building up that picture of what's actually happening in industrial biotechnology in Scotland and beyond. So of course, supporting Scottish ministers, a massive element of this role, keeping the ministers informed on progress, relevant news, facilitating events and meetings, and generally helping things run smoothly and help the minister have all the facts in advance so that they can have productive meetings, and of course, ensuring that actions are followed through. As Mark was mentioning earlier, we have a big focus on the National Plan for Industrial Biotechnology, and we have regular conversations with Mark and the team at IBioIC on how we're progressing to the ambitious 2025 targets and establishing ways that the Scottish Government can support that. So we also need to understand the wider policy landscape. So we work closely with colleagues across government to understand what's happening elsewhere and what that means for our own policy area. Working with UK government is also a key part of that, and we have regular meetings with the team at BEIS who are responsible for the UK bioeconomy strategy. Over the last year, we've established a bioeconomy network across Scottish Government, so bringing together a variety of teams who have an interest in the bioeconomy. And as you'll all be aware, it's very varied. We have representation from energy, transport, forestry, aquaculture, agriculture, circular economy, life sciences, and more. This network has really helped us to understand the policy landscape, keep us up to date of what's happening, and to facilitate connections and introductions. I think for a policy area that is still relatively newish, this collaborative approach is really key to ensure that the right people understand what IB is all about, and so that we can identify potential opportunities for Scotland. So what I haven't covered is the vast array of ad hoc activities that we get involved in: press inquiries, correspondence, parliamentary activities, data analysis, the list goes on. No day is ever the same, and it's a really varied job.
As Heather mentioned before, we work in a team called UK Industrial Strategy and Technologies. We've got a focus on emerging technologies within Scotland, and colleagues in our team look after quantum, photonics, and robotics, which makes for really interesting team meetings. We also have a focus on the UK industrial strategy and the subsequent UK industrial policies, particularly how to leverage these funding opportunities within Scotland. Our team produces a quarterly newsletter that has details of these opportunities, so if you'd like to subscribe to that, please let us know. The wider division looks after manufacturing and industries, with the overall purpose of helping manufacturing and industrial sectors to flourish in Scotland, supporting sectors to realise their full economic potential, and working with stakeholders to enhance their economic infrastructure, spur innovation, and support supply chain development. We have teams in the division who look after policy on advanced manufacturing and the delivery of the National Manufacturing Institute for Scotland, or NMIS as it's usually known, as well as leading on Making Scotland's Future, which is our program across industry, government, and academia to drive the manufacturing sector. We also have teams in the division which support specific sectors such as aerospace and construction. Our advanced manufacturing team recently published a recovery plan for manufacturing after consultation process, and they're working with a variety of stakeholders to progress the actions within the plan. This week, the team also launched the first CivTech 6 challenge, which asks, how can technology help manufacturing businesses decarbonize while building resilience and strengthening competitive advantage? The CivTech Alliance behind the challenges is a worldwide network of public, private, and third sector organizations working across civic and government tech to nurture an ecosystem which develops products and services that make citizens' lives better. Originally formed in Scotland, they focus on driving innovation in the public sector by solving challenges in collaboration with innovative businesses, and in doing so, create generations of sustainable high-growth businesses. We can send IBioIC links to any of the above should you want to know more, and please let us know if you'd like to make contact with any of the teams I've mentioned in our division, and we can make the introductions for you.
So if we have one ask from you all today, it's really to keep us informed. Tell us what's happening in your business. Tell us about your successes, the challenges that you face. It really helps us to build up that picture of what's happening across industrial biotechnology in Scotland, and enables us to facilitate introductions across government and inform the policy decision makers. I will actually be moving into a new post in a couple of weeks' time within the same division into the supply chain development programme. The Minister for Business, Trade, Tourism and Enterprise, Mr McKee, recently spoke about this work to the Scottish Bioeconomy Council, so hopefully our paths will cross again within my new role. For now, if you'd like to hear more about our team or the wider division, or would like to share information on your business or anything else, please contact Ali or our team leader, Brenda Coulter. Thank you so much for listening and having us along today, and I hope you found this interesting. Thank you.
Thank you.
Excellent. Thank you very much, Heather and Ali, for joining us this afternoon. I know how busy you are, and also I think you're being more than kind in saying we've made your jobs easier. I think actually you've made our jobs easier, because we would never manage to penetrate where we need to get to without people like you helping. So thank you very much, and hopefully you can enjoy some of the additional afternoon, but we understand you're always very busy around Scottish Government activities. So thank you for joining us. Now on to the first of our two case study speakers. So we've got Dr. Adam Ostrowski, Technical Application Specialist for Cellexus International, who's going to tell us about the CellMaker. Adam, welcome, and also congratulations to Cellexus. I believe you've just been awarded the title of Leading Pioneers in Single-Use Airlift Bioreactor Technology 2021 at the Biotechnology Awards in 2021. So well done, and a great achievement, certainly. I'll hand over to you, Adam, and look forward to hearing your case study.
Thank you very much, Mark. Yes, and indeed we did get those news relatively recently, and we are quite proud to be doing quite well. Right, with no further delay. Firstly, thank you very much to everyone at IBioIC for your support to Cellexus, which we've received in the form of a grant and many communications and advice along the way. And thank you very much for giving me the opportunity to speak to everyone here today. So I would like to present to you what we do, who we are, where we came from, and where we're going in the nearest future. And basically everything is about that we do is about lifting biological material with a bit of air, and everything's about bubbles. So to start, as soon as this decides to work, there we go. Right, here we are. I'm a technical application specialist at Cellexus. Obviously, there's many more of us than just me. We are led by Gavin Hans, who is the CEO and director of the company. everything that is technical engineering background and construction of our devices is led by Brian Wales and his right hand Craig. Everything that you would see on social media or online or any other press releases is most likely being created by Lisa Tate, who is in charge of our marketing. And we have been recently joined by Raisa Yusuf, who is taking care of our relationships with a bigger market of distributors and our network of distributors around the world. So if you ever needed to find us, we are located in Dundee. As you could see on the previous slide, we are quite a small company. At the moment, there is 7 of us in total. However, we're growing. We've got 2 positions open right now, and we're planning to expand the team further. The basis of the Cellexus is to design and manufacture the world's class bioreactors, which we call the CellMaker. As I said, we serve our customer distributor base directly from Dundee. However, we work with various other companies who help us along the way. The chassis of the reactor is made in Cambridge. Our bioreactor bags are made in the US. So we've got quite a large network of suppliers as well. The solutions we supply are ready to go, they're off the shelf, but we also pride ourselves with a bespoke aptitude to all our customers. Many of them have very unusual needs, which we strive to fulfill. And we continuously work to expand our offering. And actually, the Cellexus as a brand is not that new. It was funded in 2008 in Cambridge as a startup, which in due course has received some funding from Scottish sources, which also meant they had to relocate to Scotland. They chose Dundee as their base. Their business was progressing and things were developing. At some point, this Cellexus Limited has been acquired by the current management and became Cellexus International in 2017. And after rebranding in 2018, we are in the form that you can see us right now. As you can see here, we are a small company, and you could think that we are doing things on a small scale, but we do not. We've got a big network of distributors across the world, and we've got an active customer base not only in the UK and the EU, but also as far as the US and Canada. And we are now working on conquering markets like Australia, Korea, and China. Right, so the CellMaker technology itself is based on a patented shape of a bioreactor bag, which allows us to aerate the fermented in a very specific way. What you can see to the right is an animation of air bubbles rising from the sparger tube located on the bottom of the fermenter bag. And that provides a very special type of mixing and aeration inside the bag. Basically, all the biological contents is being lifted by the curtains of the bag and circulates in the top shelf or top side of the bag. Any cells that would try to slide down the front ramp will be immediately picked up again by the rising bubbles and returned to the most efficient part of the bioreactor. The bags themselves are very handy for multiple applications. There's a whole bunch of accessory ports that you can either sample or add stuff to. We've got several sensors in the bags as well, and we keep on working on development of additional sensors to make the use of the bag more diverse. At the moment, we are most known among bacteriophage producers and also microbial in microbial fermentation, but we are expanding further into mammalian fermentation, also with help of IBioIC. Right, so the machine itself, which you can see an example of here, is pretty small. It's a very handy, easy to use, easy to install device. Unlike many bigger systems, we can train anyone to use the system within probably 2 hours. It's relatively low investment, low footprint, easy to install, as I said, plug and play. It has very low downtime in comparison to majority of systems, because we not only use airlift system, but it's also exclusively a single-use system, which means there is very low risk of cross-contamination between individual runs. What the user has to essentially do is take out the one bag that has finished the fermentation, insert another one, fill it up with media, and they effectively ready to go within less than an hour. That removes any need for sterilization, qualification of the equipment, and again, contamination risk is reduced. The way that the technology has been designed also means it's highly flexible and adaptable. So at the moment, we've got users using them, as I said, for phage production, microbial processes. We've got a trial going on with stem cells. We've got free-floating mammalian cells on the microcarriers, and even some plant cells. So we can do really lots. Again, the system is very easy to use. We've got a bespoke software, which is very simple and intuitive. And we also really scalable, because the system that you can see here in this picture will take you from anywhere from 1 to 8 liters. And we've got a bigger enclosure, which will take your fermentation up to 50 liters. And even for the simpler applications, we can provide a single controller, which will drive 2 50-liter processes at once. As I mentioned, the technology is hugely versatile. Bacteriophage is, again, our main field. As far as I know, we are probably the only producers of bioreactors, and definitely single-use bioreactors, that directly engage with bacteriophage producers. Some of you might know there's a network called Phage Directory. It's originally a newsletter and then a website, and now a whole community that has been started by a graduate student in the US. It is a center point of the whole bacteriophage research world, essentially. And we are very proud to be part of that environment. We already had people coming to us asking for our machines, saying, "Oh, I heard about you from such and such through Phage Directory." So it's really, really cool environment to be in. And again, from bacteriophage, it's very easy to reach towards microbial fermentation. Our systems have already been used with standard bugs like E. coli and Salmonella, but we also went into slightly more complicated routes like Pseudomonas, Staphylococcus, and even Yersinia. And finally, as I said, we continuously develop, and we've even converted one of our standard units into a new model, which we call low flow, and that is specifically designed to enable mammalian cell culture with minimum shear stress, with good conditions for the mammalian cells to thrive, minimize the foaming, which is inevitable in any airlift system, and provide people with means to upscale otherwise difficult to scale production of mammalian cells with maximum yield of bioproduct. To give you an example, or a few examples of what we've been doing so far, here's a application note, or parts of the application note done by a Belgian customer in it's a Catholic University in Leuven, laboratory of Professor Rob Lavin, where they are developing bacteriophage therapy or treatment against agrobacterium to protect certain plant species. What they normally would do is cultivate their cells in a traditional shaken flask, inject the phages at appropriate stage in the bacterial growth, and get some phages at usually slightly unpredictable process, huge differences from batch to batch, from day to day, from flask to flask. The average result would be about 10 to the 9th of phage particles per milliliter of culture. We came in with our reactor, and basically within single experiment, we managed to increase their production for over a degree of magnitude. Another example slightly closer to home from University of Dundee, Dr. Jean-Christophe Bourdon specializes in hybridoma cell lines and uses them to produce antibodies. Usually that is done in very small batches, lots of flasks, lots of manual handling. What he managed to do is to, using our reactor, move his scale from 300 milliliters per vessel to 2,000 milliliters per vessel, and not lose the yield in significant degree. However, the ease of manipulation of the system was hugely increased. So taking the production from about 5 micrograms of antibody per vessel to 26 micrograms per vessel was for him a huge improvement in the handling of the process. And finally, what we do right now with IBioIC's help is a project together with University of Alberta Dundee, where I work with Dr. Sean Brown and Dr. Scott Cameron on proof of concept project to show that CellMaker is an ideal vessel to grow Chinese hamster ovary cell lines with aim of producing monoclonal antibodies. What we want to do there is basically show that by use of CellMaker, you can save time and effectively money over a traditional shaken flask culture. And we do this because the Chinese hamster ovaries line is essentially the workhorse of mammalian cell biotechnology. And at the moment, we don't have data to prove that we indeed can do it. We know that we can do it, but this is one of our ways to show that it is indeed true. The aim of this project is to produce a peer-reviewed white paper to showcase our technology. And that is essentially me. Thank you very much again for this opportunity. And if you would have any further questions, you can reach us online, or I'll be here throughout the afternoon. So I look forward to hearing from you.
Thank you so much, Adam. Thanks to Adam Ostrowski for presenting the first of our member case studies and introducing Cellexus and the CellMaker, which looks like great technology. For those of you that I haven't met, my name is Caroline Cuney. I'm Senior Business Engagement Manager working with the membership at IBioIC, and I'm here to guide you through the rest of this afternoon's events. So we'll just move swiftly on next. I'm pleased to introduce Dr. Leonardo Magnesci, who's Head of Molecular Biology at Ingenza. He was going to present our second case study on enhanced biomanufacturing of industrial products and biologics through integrated synbiotechnologies. Welcome, Leonardo.
Thank you very much, Caroline. I'm going to share my screen. right now. Right, so thank you for giving us the opportunity to present Ingenza at this IBioIC Member Summit event. I'm just going to give you a very short introduction about the company, and then I'm going to move into a couple of case studies just to give you a flavor of what we have achieved with our customer. So we are a world leader in applied synthetic biology, based just outside Edinburgh at the Roslin Innovation Centre. You can see a picture of the team here in front of the new building where we moved roughly a couple of years ago. And we specialize in the design, development, and manufacturing of chemicals, biologics, pharmaceuticals, and biofuel products. We were established in 2003 as a spin-out company at the University of Edinburgh, and we have a multidisciplinary team, proprietary technologies, and very flexible collaboration and business models. So one of the things that differentiates Ingenza from other biotech in the same space is the fact that we not only have a molecular biology department, but we also have a fermentation team and chemistry team that sort of help tackle the challenges from our customer altogether. So we have genetic engineering and fermentation capabilities on more than 10 different microbial hosts. You will see some of them reported here in the slides. So we have worked with E. coli, the workhorse for most of our partner, but we also hijack Bacillus protein expression system for especially secretion of protein, Pichia pastoris and Saccharomyces of yeast models, CHO mammalian cells, and then other less famous microbial hosts like Corynebacterium, Pseudomonas, and Streptomyces. We have proprietary DNA assembly technology, and this is used for the optimization of recombinant protein expression, for example, where we typically test different codon modification, promoter, and regulatory elements. But we can also perform evolution and mutagenesis program for specific microbial hosts alongside construction of specific metabolic pathways. Our fermentation team, as you can see here in the map, we have scaled up and developed fermentation processes a little bit everywhere in the world, really. We can perform cell banking and scalable fermentation process development from 5 to 20 liters in-house, but with partners, we have scaled up our processes to 2.6 million liters in the US, especially. Our chemistry team is responsible for the analytical and synthetic chemistry part of things, but are also engaged with downstream processing, troubleshooting, recovery, purification, and analysis of the different recombinant protein and metabolites that we produce. We also have a strong QMS department in-house and electronic lab books. So in this slide here, just capture some of the areas that we operate in. So we typically work with some manufacturers that want to move away from petrochemical sources and establish a more like a greener route. But we have also collaborated with, say, academics to bring their novel antibiotics to the market, some agritech company for food productivity, healthcare quality. We are working on reducing emission of greenhouse gases, and we also provided superior microbial strain for the production of sustainable liquid fuels. So on top of the customers that you see here in the slides, we have an extensive network of academic collaborators, both in the UK and in Europe. So some of the information and data that I'm going to present in these slides are actually derived from an IBioIC PhD program in collaboration with the University of Glasgow. So at Ingenza, our motto is we basically get what we screen for. So a lot of the time and efforts from our chemistry department is linked to the generation of specific screen that basically generates a lot of power in the selection of the variability that we can generate with the molecular biology. So in specific case here, a couple of them. For a leader in the perspex manufacturing, we developed biosensor for the detection of the building block of durable bioplastics, where you basically have the higher the fluorescence, the higher is the capability of the strain to actually produce this building block. We have collaborated with biopharma in the US. Here, we basically help them in generating a sequence of their biologics that was more efficiently translated. And we have done this by either fusing the product with a specific tag that generates this red color. But we have also in recent years developed an in-house codon optimization algorithm. So as you can see, when you compare the same protein sequence that was codon modified by, say, GenWiz and Twist, so two of the major providers in the industry. With our algorithm here, the boost in expression of the recombinant protein is quite evident. We have worked with some companies, for example, to move away from antibiotic selective marker. Here we have developed a specific auxotrophic selection system. And we have also helped some nascent companies, for example, Amprologics, to bring some antimicrobial to the market. They're currently at the tox study now. This was possible by generating both a cell-free extract in vitro transcription translation, but also a very effective screening for the biological activity of these antimicrobial peptides towards different microbial hosts. With something that I'm going to talk about later on, this visible platform here, we're basically generating fusion protein of a novel fluorescent marker with our protein of interest, and this allow us to select via both FACS, so fluorescent assisted cell sorting, or in the supernatant, basically by looking at the fluorescence, then we can quickly identify strains that express and secretes the protein of interest. And then last but not least, we have worked with consumer goods manufacturer, where we develop assay for the evolution of fatty acid modifying enzymes. So as you can see here, our library, that's a negative control, red color or blue color. The positive control, which was the competitor enzyme, generates either a dark blue or a yellow color. And when we generated the library of interest, you can clearly spot here some of the candidates and best performer from the whole library. So if I now talk a little bit more in details about this visible platform, this was specifically developed through an IBioIC PhD program. And what we did here in PICA was to basically use this ILOV domain, which is present in plant protein, but is basically ubiquitous in a lot of different organisms. This domain can be modified, and if slightly modified, can fluoresce upon excitation of blue light. Fuse it with basically a linker that can be cleaved scarlessly, and then put your target protein at the C-terminus of this fusion. So this system basically allows visual monitoring of both upstream and downstream productivity, and also the stability of the strain, because problem with PK is that a lot of the strain will lose the copy. So if you see a drop in fluorescence, then you know that your strain is not stable, so you can go back and identify a different candidate. It's free of third-party IP, compatible with FACS, as I showed you previously in some of the example, and can mask the toxicity effect of some of the targets. So in the example that I'm going to give you in the next slide, specifically here, we have used this visible platform for the production of a novel antimicrobial peptide, which is effective in a nanomolar range against superbugs. So antibiotic resistance, MRSA, and so on. So this antibiotic or antimicrobial peptide is 15 times more effective than the market leader. And although the academic was able to produce this specific protein or peptide in the microgram per liter, we have developed a whole process allowing now production of grams of the biologics, and therefore competitive cost of goods. We have also managed product formulation and preclinical toxicology through third parties. So we have identified and established good collaboration with those to bring the antimicrobial peptide to the market. And the way we did this was transform PK pastoris, which is a host usually used for protein production, allow tolerance to the biologics via visible, because as I said, is masked toxicity, and then generated a massive library using both random integration, different codon modification, and selection through fluorescent assisted cell sorting. So as you can see here in this graph, we have most of the population localizing in this area of the fluorescence, and then we design a window that captures less than 2% of the population, and then candidates were further evaluated. So by doing so, we have established a 20-liter scalable fermentation process. You can track here with this yellow color the purification diffusion. And then if you can look now at the top right figure here, you have the pre-cleavage target fusion. Post-cleavage, you will have both the tags, so the ILOV and the target, and then following purification, you have more than 95% pure target. So this system has allowed us to generate a lot of the biologics needed for tox studies, but also isolating the best performer in within a mix of like several million clones of Pichia pastoris. We currently actually recently use the same system for secreted protein. Here we were contacted by the University of Oxford, some of our collaborators. They were producing some vaccine component in mammalian cells, and they wanted to move away from this costly microbial host. So we developed a system in Pichia using the Visible platform that again will produce the ILO fusion to the RBD, which is the target or antigen, and then following cleavage of the ILO molecule, you will have the target to high purity.
Hi, Leonardo. I'm so sorry to interrupt. Just to let you know that your time is actually up in terms of presenting, and we do need to move on. So if we could see your final slide and just
sure. Yeah,
thank you very much. Perfect, thank you.
That was basically it. So my last slide is, well, we are on the market since 2003, multidisciplinary team, and we have some proprietary technologies that could solve some of your problem. So if you want to get in touch, just drop me an email and I'll be happy to answer your question. That was it. Thank you.
That was great. Thanks, Leonardo, for that excellent presentation. That was really good. Yeah. Moving on, I'd like to now introduce you to some of our member pitches. So this is the first of 2 rounds. We've done some quick fire member pitches where we'll hear briefly from 8 of our members in round 1. We're really pleased that our newest member, Beckman, Coulter, are taking part. So we'll just go ahead with those 8 pitches now.
Hi, my name's Chris and I'm the Customer Operations Manager at Amici. Amici is a Glasgow-based company and we've been around for 17 years. We work with biotechs all around the UK and in the US. We have around about 150 biotechs that use Amici in the UK, with thousands of users using our platform daily. We have a good number of customers in Scotland, including at the various science parks in Glasgow, Edinburgh, BioCity, and even as far as Inverness. Now, all of our customers want to spend as much time as possible in the lab or supporting the lab, and as little time as possible when it comes to purchasing and managing orders. And our job within Amici is to help them do that. All of our customers use our platform, where you can log on to one platform, search for whatever products you want to buy, add it to your basket, get approval, and automatically place your purchase order. Now, on our platform, we have millions of products available, from lab consumables to components to chemicals and media to equipment. And it's one place for you to raise all of your purchase orders. Behind the scenes, we have a team of around about 60 people that work at Amici, and our job is to help you, whether it's saving you money on the products that you buy, managing your purchase orders for you, or providing a whole host of operational solutions, from setting up simple stock lists to managing your equipment and service contracts. If you'd like to know any more information about Amici, please get in touch. Thanks.
Have you ever felt insecure about strategic decision making? Have you ever needed a carefully prepared strategy but hadn't the time or the know-how? Have you ever wondered how to get access to new customers and cooperation partners? Hi, my name is Tobias Kirchhoff, Senior Consultant at BCMP Consultants in Germany, and if these kind of questions coming up again, we will have solutions for you. We offer you 3 things. First, security. You get decision-making reliability for decisions about markets, cooperation partners, and investment opportunities. Second, ability. You are able to start implementation of a new market entry or a setup of a new business by a fully worked out strategy. And third, velocity. Your innovation activities will be accelerated by finding new cooperation partners and customers, either by direct support or through DCP. In our business unit BCMP strategy, our services include specialized market insights for technology-driven products, services, and companies. Either for your individual market analysis, for scouting activities, your business planning, or your commercial due diligence. In our business unit BCMP Connect, we offer you access to new potential customers by our huge industry network. And this network you can also meet at our ECP. The quality of the services bases on 4 pillars. Our specialization on biotech, chemistry, nanotech and pharma, our huge network, our interdisciplinary and our close collaboration with you. In the past, we worked successfully together with corporates like Symrise, with investors like the European Circular Bioeconomy Fund, but also with LMEs, startups and institutions like CLIP. If you feel insecure in the future about decision making, contact me and we will find the best fitting solution for you.
Hi everybody, it's great to be here today to take part in this networking event, and I'd like to thank the organizers for this opportunity to introduce myself and Beckman Coulter flow cytometry to you. My name is David Kivlikin, and I joined Beckman Coulter Life Sciences at the end of last year as the flow cytometry sales representative covering Scotland. My contact details are on the screen, or feel free to connect with me on LinkedIn or Twitter. Today I'd like to talk to you about the Cytoflex family of flow cytometers. They are the first commercially available flow cytometers to use avalanche photodiodes, or APDs, instead of classical photomultiplier tubes. APDs give high resolution and sensitivity, as well as allow gain-independent compensation. The Cytoflex's small size means it takes up less space on the bench and can even fit inside a hood. We have a variety of configurations from 1 laser, 4 color, up to 6 laser and 21 colors. They are also available with 96 well plate loader options, and our site expert analysis software is intuitive and easy to use for new users and comes with unlimited site licenses. I'm especially excited to introduce to you the newest member of the Cytoflex family, which is the SRT benchtop cell sorter. Built on the technology of the successful Cytoflex analyzers, the SRT is a compact, easy-to-use sorter that can be configured with up to 4 lasers and 15 colors and has 4-way mixed mode sorting. It can also sort into 384 well plates. I hope you find this information useful, and if you would like to discuss further, please feel free to get in contact. I'll be around the networking later. Thank you very much, and bye-bye.
BPES connects the industrial biotechnology industry to market-leading innovators of specialist equipment. Our team of technical sales and application specialists are astute to the needs of our customers and provide effective and bespoke support from order to delivery, installation and aftercare. We work with 16 suppliers across the biotech, biopharma and cell and gene therapy sectors. 3 industrial biotech suppliers with proven solutions in this IBioIC network are Solaris Biotech, ABBA and MTI Bioscience. Solaris Biotech specialise in the manufacture of fermenters and bioreactors for R&D and production. With a volume range up to 30,000 litres, Solaris's industrial fermenters offer completely customisable and highly automated solutions tailored exactly to your specific needs. Bringing offline measurements online, ABBA's capacitance-based technology allows real-time, accurate measurement and control of live biomass concentrations in fermenters at both laboratory and industrial scale. MTI Bioscience provide continuous thermal sterilisation systems for heat denaturation of microorganisms. This established technology provides very gentle, quick heating. It is validated in real time, providing you with constant quality that is easily scalable. We would love to hear about your projects as well as any questions you have about how our product range can support your process. So please get in touch at info@bioprocess-eng.co.uk.
Hi, my name is Elaine Baxter. I'm founder and director of Boutique Innovation. I'm an innovation coach and specialist with a background in chemistry. I worked for Procter & Gamble, Gillette and Coty for 15 years, ultimately delivering their first innovation acceleration programme in the UK. Who do I work with? I work with a range of clients from one-woman startups to large corporates and across many sectors in the last year alone, having worked in energy, utilities, food and drink, pharma and construction. I work with all the functions, everyone from marketing and sales to R&D, and I've even worked on organisational change. How do I work with clients? I take my clients through a series of methodologies and workshops which I've developed over the years, where we go from concept to ideation and prototyping and test over a series of engagements together. I like to initially connect with people in an informal way to understand what their needs are and figure out the scope of a project. And from there, we can decide whether or not we want to work together, anything from a 1-hour engagement to a series of months. So please feel free to reach out informally and let me understand whether or not I can help you to deliver better outcomes for your business.
Hello, I'm John Henderson and I work for Brightest, a leading exponent of whole process design for the process industries. Brightest's technical facilitation offering helps multidisciplinary teams of all sorts generate value through enhanced whole process understanding, gaining new and deeper insight of the science driving their technology, and broader shared understanding of the business context and criticalities against which the technology will be assessed. This is underpinned by a suite of innovative business tools and techniques for knowledge capture and sharing that extends to meet the particular needs of bioprocesses. Brightest bioprocessing tools can help troubleshoot or understand complex whole bioprocesses, tracking process materials and assessing the impact of variability in the process on quality, operability, productivity and safety. At a higher level, we can help map out process techno-economic calculations for what-if scenarios and target setting for new technologies. And our multi-criterion decision support tool lets you visualize uncertainty, allowing risks to be identified and managed when planning investments. Speaking of risk, Brightest's scale-up risk assessment methodology deals directly with a vital stage on the road to commercialization that has been a stumbling block for many a novel bioprocess. Brightest's specialist technical facilitation service maps directly onto some of the most frequently asked questions on both sides of the valley of death, which innovators in industrial bioprocessing must cross if their hopes for commercial impact are to be realised. If you'd like to discuss how we could help you, I'd be glad to talk during the networking session.
Hi everyone, my name is Matt Connolly and I wanted to tell you a little bit about 5-inch Software. We're a software consultancy who provide end-to-end managed projects for the life sciences and biotech sectors. We have a strong domain knowledge from years of experience working on scientific instruments in laboratories ourselves, as well as alongside other bench scientists. And this means we can speak your language, understand your problems, and deliver the right solutions for your business quickly and easily. Our clients vary from startups to multinationals, with our projects ranging from instrumentation software through to automated business processes or producing customer-facing web applications. For example, we recently had the pleasure of working with Dynaval, the winners of the 2021 IBioIC Scottish Edge Award, to help productise their semen motility device. We developed a desktop solution to provide instrument control, data processing, and a cloud-based analysis and storage system for their instrument using an agile approach. So if you'd like to hear more about the sorts of things that we can do, please meet up with one of the team at the networking session or follow us on LinkedIn. Thank you.
Hello, I'm Stuart Little, and today I'll be showing how JMP can be used to translate your data into insight and understanding for your processes, and how in doing so, this can reduce the time it takes to get your products to market. JMP is an interactive and visual tool for data exploration and statistics. In this example, JMP has been used to successfully model a protein expression process using design of experiments. The outcome of this work is a profiling tool, which allows us to interactively explore how changing any setting in the reaction might affect cell growth. This allows us to see what the effect is from the current ones to those that might give us optimal growth
but also perhaps the most robust settings for manufacture. This is a very common approach across the biotechnology industries. As an example, Lonza are using JMP to reduce the time it takes to bring new manufacturing processes online. In one case, they used design of experiments methodology to nearly double the product yield of their product, and at the same time halved the number of experiments it took to achieve a production-ready process for their biological product. Thanks to the effectiveness of these techniques, statistical methods using JMP are now thoroughly embedded into Lonza's biotechnology workflows. There is plenty more information about this case study and many others at our website at jmp.com. And if you wish to have any more information, please feel free to reach out at the email address shown on the slide. Thank you for your time.
Thanks so much to all of the quickfire speakers. It was great pitches and gives a flavour of some of the breadth and scope of the range of work our member organisations are involved in. We'll now take a short comfort break and reconvene at 3:40, and with another set of member pitches to hear then. Some information on our funding will be shown during the break. We look forward to seeing you back in 6 minutes at 3:40. Welcome back, everyone. Thanks for returning after the break and this lovely afternoon. Without further ado, I am pleased to introduce our second round of quickfire speakers. Quickfire member pitches. We'll now hear from some more of our members.
Hi, my name's Mary Rudkin and I'm a patent attorney. I work at a firm called Marks & Clark in Glasgow. Marks & Clark is an intellectual property specialist firm with a worldwide network of offices, and across those offices we have patent attorneys, trademark attorneys and also intellectual property solicitors. We have 3 bases here in Scotland. I work in our Glasgow office, but we also have bases in Edinburgh and Aberdeen. My own background is in chemistry, and I did my degrees at the University of Edinburgh. I have a particular interest in sustainable chemistry and the circular economy, and in the industrial biotech sector, I've assisted a number of emerging biotech companies in a variety of technologies, including new processes for converting waste food materials into useful biodegradable products, and also the application of bacteriophage technology for decontaminating foods of microorganisms that might lead to food spoilage. I'm here today because I'm just really keen to hear about what's been going on and the latest developments in this sector. I'd also be keen to answer any intellectual property related queries the members might have. And as a final point, it would be good to hear if there was any particular topics that the industrial biotech community would like to hear about from IP specialists in the future. Thank you.
I am Dr. Eve Hanks. I'm a veterinary surgeon, research scientist, and I'm CEO of MyRNA Ltd. And we are a veterinary diagnostics company using biomarkers from biofluids, such as blood and urine, to revolutionise early disease detection. Using this pioneering technology and combining it with our bespoke artificial intelligence model, we plan to provide accurate disease detection in a range of species and a range of disease types. Our mission is to reduce disease through targeted, evidence-based research and innovative partnerships, and thereby improving animal welfare, which results in a cascade of ethical, environmental and economic benefits. Currently, we're about to commence research and development into Johne's disease in cattle. Early diagnostics for this global, incurable disease are currently unavailable and would provide critical economic and environmental advantages to the agricultural sector. Production-limiting diseases make agriculture unsustainable in its current form, with Johne's disease alone costing the Scottish economy £13 million per annum. Milk production from a Johne's infected cow comes at a cost of an increase in greenhouse gas emissions of 25% per unit, and in meat production emissions are 40% higher. We are looking to engage with academic and scientific experts in production animal disease, environmental impact and food waste reduction, and we're also interested in third sector views and policy. And finally, we would appreciate any signposting towards appropriate funding routes as we raise our first round of investment. Thank you.
Hi, I'm Jen van der Hoven, Director of the National Horizon Centre. Thanks for having me here today at the IBioIC summer event. I'd just like to take a few moments to talk to you about what the National Horizon Centre is and how you can partner with us. So the National Horizon Centre is a centre of excellence for the bioscience industries. And through our delivery model of research, partnerships and training, we bring together industry, academia, talent and world-class facilities to create real-world impact. And together with our partners, we pride ourselves on discovering diseases earlier, developing novel treatments and delivering life-changing medicines to those in need quicker, safer and more affordably. There's 3 core pillars to the NHC. The first being research, the second being partnerships and the third being training. And across those 3 pillars, we have 3 core research strengths. The first is disease-specific research, where we're looking to understand the underlying biology of diseases and detect them earlier in patients. So, for example, working on cancers, Alzheimer's, and asthma. The second is biomanufacturing, where we're looking at food technologies and then also biopharma, so developing and optimizing biotherapeutics. And then the third is the digital technologies that underpin those other two core research areas. So we've got expertise in bioinformatics, data analysis, and virtual reality. A few very quick examples of the partners that we're working with to translate basic research into more applied research are CPI, Fujifilm, Unilever, Croda. We've got a strategic partnership with the NHS. We're also working with Quorn. And in terms of our training, the NHC is actually a national training centre for advanced therapies, and we provide continual professional development training in advanced therapies, vaccine manufacturing, bioprocessing, digital technologies, and that's all underpinned by using our world-class virtual reality training suite and also hands-on in our amazing state-of-the-art labs. We're always wanting to find new partners to work with, whether that be in the research or the training, so please get in touch. Thanks.
Hi, my name is Graham McDonald and I'd like to introduce my business, NZC 2030. I retired last year as Managing Director of ENVA, or William Tracy Ltd as it was formerly known, and we were Scotland's most innovative waste management organisation in that we didn't operate landfill sites, so the focus was very much on collecting materials, processing them, creating markets and finding alternative uses. And this is the approach which I've currently taken with the current project I'm working at, supported by IBioIC and University of West of Scotland, where we're using organic waste as a food source for mycelium growth to create mycelium insulation blocks and potentially other materials such as a substitute for polystyrene packaging. As you'll be aware, there's a lot of societal changes now in terms of circular economy objectives and carbon reduction, and we very much want to move into this, see this as an opportunity to develop new business models. So this is the first project we're looking at, but I'm very keen to collaborate and work with other projects within that field with an aim of creating business growth and manufacturing jobs across Scotland. Thank you.
Hi, I'm Kenneth McGregor from the Scotch Whisky Research Institute. We represent approximately 90% of Scotch whisky production, and I'm here today because I'm interested in collaborating with technology providers that can utilise our biomass streams from the production of Scotch whisky. So there's four areas. There's draft, so that's the residues from the malted barley that contain protein, starch and ash. We have pot ale, which is the residue from the first distillation stage, and that's protein and carbohydrate and some copper in there. We have spent lees, which is the residues from the second distillation stage. That's kind of dilute organic acids, alcohols, and has high levels of copper. And the last is the pure biogenic CO2 that we have from the fermentation stage. Please change slides. So a medium-sized distillery in Scotland would produce quite a sizable amount of co-product, and at the moment we have sustainable means of dealing with these co-product, but we're always interested in increasing our sustainability. We're particularly interested in collaborating with someone that can use spent lees and biogenic CO2. We're currently involved with IBioIC and Zero Waste Scotland, examining technology applied in series. So this project involves the removal of protein and carbohydrate from pot ale using two technology providers, and we've got plenty of time, to dedicate time to other projects.
Hi guys, I'm Douglas McKenzie, Chief Executive of Untitled Ltd. But today I'm here to talk to you about our new spin-out, which is called FICO-F. And FICO-F has been developed basically to exploit the results of the Scottish Government funded ASLI and MBio projects. And we're dedicated to making algal production at industrial scale economically viable. Now, microalgae and the photosynthetic production of microalgae is one of the best opportunities in industrial biotechnology, partly because of the wide range of valuable products that you can make with algae, and partly because of the simplicity of the feedstocks. However, it's limited by the high cost of production, and most of that is down to the cost of light. Now, virtually all industrial production of microalgae at the moment uses sunlight, which obviously not particularly great thing for Scotland, but equally it's pretty much a dead-end technology, so we can't reduce the costs, and it introduces a whole lot of problems as well. However, artificial light, the cost of that is decreasing rapidly, and over the past 10 years it's fallen by about 2 orders of magnitude, partly because of LEDs, and partly because of renewables and the decrease in cost that has come about because of that. Next slide, please. So our commercial strategy is to access low-cost electricity, and there are actually opportunities to get electricity at much reduced cost, sometimes actually for free, and also to access fermentation CO2, particularly from distilleries, and use nutrients from anaerobic digestion, which is becoming increasingly used in distilleries as well. And then to make money through both selling our technology, and particularly our systems technology, but also acting as a contract manufacturer of sustainable algal biomass and products derived from that. And so we work in partnership with people who can supply us with the feedstocks, often because these feedstocks are a problem to them, and turn them into valuable products. And that's FICO-F. Thank you.
Good afternoon, everybody. My name is William Clark, and I'm here today to give you a very quick introduction to Zero Waste Scotland. So very broadly, we work to support Scotland to use products and resources more responsibly, with our focus on where we can have the greatest impact. And the diagram here shows you some of the themes within Zero Waste Scotland, but also illustrates that circularity is at the core of everything that we do. So as a very brief introduction, we work across food systems, energy and bioenergy, built environments, the bioeconomy, materials and services, but we also work with government to provide evidence to develop better policy. We enable the green recovery through our work with the skills and education sector. We are working to support the government targets around net zero through programmes such as the carbon metric. And I would also highlight that communities, there are a number of community facing elements in the work that we do. There's a QR code in the bottom right hand corner, so you can go and visit our website and enjoy this information in much more detail in your own time. However, slide please. There are some programmes that I really wanted to highlight in today's discussion. So the Circular Economy Business Support Service offers one-to-one expert consultancy for you to help identify new growth opportunities for small and medium-sized businesses. There is also the Circular Economy Business Network, which is focused through sharing and building upon good practice in order to lead transition towards an advanced circular economy for Scotland. Probably the key one here for discussions today is the Circular Economy Investment Fund, which offers grant funding to SME businesses who are helping to create a more circular economy. And last but not least, the Circular Economy Innovation Workshops, which are really aimed to inspire circular economy thinking and develop and build and refine ideas. Thank you.
Hello, everybody. Just to wrap up this really great section of quickfire talks, we've got something slightly different. I'm going to introduce Dr. Joe Price from the University of Sheffield. You may know the University of Sheffield is one of our IBioIC's academic partners through the N8 Research Partnership. Joe's developed a piece of technology that's optimized for next generation microbe engineering called Evoluta, and he'd like to tell you about it. Joe's currently on the iCure program, so if you have any feedback on his technology, please do get in touch and let him know. Thanks very much, and over to you, Joe.
Hello, thank you for having me here today. My name is Joe Price and I'm based at the University of Sheffield. There, my team has developed a new technology that we've called the Evolutor. The Evolutor is a device that allows us to automate and enhance how we optimize microbes by exploiting the natural power of evolution to a new level. With the innovations of the Evolutor technology and with the expertise of my team, I believe we can offer substantial improvements to the productivity of microbes and subsequently to the output and financial viability of precision fermentation production streams. We can more fully optimize a vast range of microbes, and we can improve many key traits like tolerance to inhibitors, growth rates, production yields, and the ability to more efficiently utilize new feedstocks. We can also do this in a way that complements and builds on top of other microbe development techniques to really help extract the absolute maximum value from a microbe's performance. At the moment, I'm undertaking market exploration through the Innovate UK iCure programme with the aim to use the Evolutor's technological innovations to commercialise a next generation microbe optimisation service. If you use microbes in your manufacturing and fermentation processes, I think that this Evolutor service would be extremely beneficial to help maximise your fermentation efficiencies and outputs. I would love to hear from you and chat more about how you think the Evolutor could be best placed to benefit you. My email address is on the screen, and you can also find me on LinkedIn, and I'm in the chat here today. Thank you very much.
Thanks so much, Joe, and thanks to all our quickfire speakers. I hope that was useful. Thanks to everyone that contributed to the pitching session. I'd like to now invite people to attend our optional breakout discussions. So we've got 4 rooms encompassing business engagement, projects, funding, and skills and training. So attendees, you'll all be randomly allocated to a breakout room unless you've indicated your preference in advance. Each room is facilitated by IBioIC staff. We'll be discussing the same sort of broad questions. Sessions aren't being recorded, so feel free to have open discussion. You'll receive a 5-minute warning and then a 60-second warning that the rooms will close. So that should be about 20-25 minutes, at which point I'll see you back here. Thanks everyone for taking part in the breakout discussions. I hope you enjoyed some good group conversation at this social event. We certainly did. We've now got an opportunity for one-to-one meetings before the close of the meeting at 5, and they're on meeting module. But firstly, can I say thank you? I'd like to round up the event with a vote of thanks to everyone that took part today, to all of the speakers and the presenters, including the member pitches, and to our event partners, Lux, including Melissa, who's been running everything behind the scenes today, and also Ian at Vision and Andrea R. Neil, live production, who've supported all of the tech for this event. Thanks to the IBioIC colleagues as well, especially Lorna Watt for coordinating the whole event, and all the facilitators and note takers that took part. And finally, thanks to all of you who have attended today. Hopefully see you at a future IBioIC event, hopefully in person at some point. For those taking part in the next session, we'll have a short 5-minute break. The networking will start at 4:30, so that allows people that have registered for meeting module to go get ready for the speed networking. So it just leaves me to say thanks so much for taking part.